Good morning. I'd like to welcome you to our second webinar of the Charting the Life Course and Interdisciplinary Blueprint webinar series. The format of the call today will be one TED-like presentation from Roz, and then we have two additional presentations from Dr. Emily Oken and Dr. Maureen Black. So we will start with that presentation and then go into directly into the other two presentations and leave 5 minutes for questions in between. So thank you, Dr. Rosalind Wright, for joining us today, and we look forward to your presentation.
So thank you very much, Heidi, for that introduction. And I'm so excited to be here today. And what I'm going to be discussing is talking to you about my own journey into complexity science and the promise of the exposome in that context of that work. So when I talk about complexity science, it's the study of complex systems that are dynamic, unpredictable, and multifactorial, multi-level. And these patterns emerge over time, and that's why we talk a lot about a life framework, which I'll be talking about as well as the challenges we faced in our own work. And it really focuses around disentangling what begets asthma. And I've been very interested in that because I am an adult trained pulmonary physician as well as a public health researcher. And in my clinical work, I've been taking care of patients at the right-hand side of this, the life spectrum, when they have end-stage disease by and large, COPD. And I got very interested as a clinician in how far back do I have to go to really have an impact on putting my patients on a better trajectory, having better health and better longevity. And it really took me all the way back to the roots of it, all the way back to pregnancy, which you see on the left-hand side of the figure here. And our focus is really, initially, there was a lot of work in asthma, and my own work was interested in the genetics of asthma. And as you can see here, what we've learned with not only asthma, but other chronic diseases, the predominance of disease burden is not caused by genetic factors, but it's really primarily environmental factors. And that is the challenge. How can we comprehensively assess the environmental factors that may be driving some of the chronic disorders that we see, including asthma, lung growth, and development over the life course. Now, asthma lends itself very nicely to a life course approach as well, it turns out, and that's very fortuitous for me as a researcher. It has its roots starting in early life. It's largely environmentally driven, and it has different trajectories over the life course that depend on ongoing exposures as well as host factors. And that's not just genetics, but you can see that things like sex, girls and boys have different trajectories over time. And actually girls start to have their onset around the time that they're heading into their reproductive years, and that has implications. And then you have ongoing inflammation and remodeling, and that asthma is the biggest cause of that factor going on, and that's a determinant of lung growth and development and what happens to you over your life course. So how do you study that in a life course type of framework? And so what we've developed over the last couple of decades is this perinatal environment and developmental studies, the PEDS research program, and that we stood up a number of prenatal longitudinal cohorts, and you can see them here over the time that we had there, and each one has about 1,000 people in it, and I'll be presenting results from the different studies as we go through the talk today. And here was our challenge. We sat down and started thinking about, well, what do we know about asthma? Now this is, almost 20 years ago, so bear with me. And we said, well, what are the key driving factors that we think that we understand? And we want to measure them as comprehensively as possible. That doesn't overburden our staff and or our participants. And so we focused on a lot of the traditional measures, the home environment, indoor allergens, outdoor air pollution, chemical exposures like tobacco smoke, and diet and nutrition. And one that hadn't been looked at a lot, but we were very interested, how does the social environment get embedded? And that was through psychological stress, as shown here. And we did this in very diverse cohorts. And what we found initially was, yeah, lo and behold, we could see an independent effect of stress if we measured it with just a life course or a life event measure, and we created a continuous exposure measure. And you could actually see that there was an exposure response relationship with the increasing number of life domains that people were experiencing negative life events. And then that, as you see the little guy running over the hill, as you anticipate that as you experience more and you overwhelm one's ability to cope, that's when you're going to start to see effects of stress, both behaviorally in anxiety and depression, but also physiologically embedded by disrupting these key regulatory systems that are pictured below: autonomic nervous system, immune function, HPA axis, oxidative stress. We look at them one at a time in a reductionist way, which is traditional. But what we really want to get to is how do you look at that in an integrated way? That's our challenge. As we went along and we repeated the measurement, not only in the prenatal period, but over the early childhood period, we started to see that there were some host factors that contributed to when they would be at highest risk. And you can see here, just looking at child sex, girls and boys are more vulnerable at different developmental windows. Boys in the blue circle are the prenatal stress seem to have a bigger impact on its relationship to respiratory outcomes. And girls, they got the hit postnatally. And you can see through the references, you can drill down a little bit more in this. The other thing we learned is these kids aged, and about age 6 to 7, when we could do lung function testing on them, you could see that there were cumulative effects of the stress. So kids who were particularly vulnerable and had lower lung function if they experienced higher level stress in both of those time periods. So there we learned you needed to have repeated measurement over these different windows and look at cumulative effects. Well, we found that timing was even more critical when we wanted to look across in developmental windows where there's rapid changes. And it's just like the key to comedy, timing is is key to life course research, and if you want to find sensitive windows in development in particular. So here's a graphic that just shows the rapid development of the lung system in utero on the left upper hand graphic here. And so there's so many changes that are well orchestrated, well timed, and ideally what you want to do is not just randomly pick when you want to measure one point in time exposure, but boy, would it be great if you could measure every day. But of course that's expensive and it's actually burdensome, so you don't necessarily do that. But how can we, as shown in the lower part of this graph, develop methods where we could get a measure over the course of the day? And if you look at 4 or 5 different people as mapped here, they have different exposures over the course. And those who have particular increases in these developmental windows may have different manifestations clinically. Well, one way you can do that and reduce burden is to actually use Geomarker data. And this is something that's developed over the last several years as a very useful tool in environmental health research and exposure assessment. And basically what you do is you take an address, you convert the address, coordinate it to the map as you see on below. And if you can do this to someone's place of where they live, their home, where they work, where they go to school, whatever you might be interested in. And then you link that spatial location to other existing data sets to assign exposures. For example, you can do environmental pollution exposures, and you can do that on a daily basis over the course of whatever interval of the life course you're interested in. In this case, we were interested in prenatal. We worked with our colleagues at the Harvard School of Public Health and the Icahn School of Medicine at Mount Sinai to use satellite-based hybrid models using integrating satellite data with ground monitors, as you see on the left-hand side of this graph. And you can get daily air pollution exposure levels. That's great, so powerful. And now you can use data-driven statistics to let the data tell you where these sensitive windows are, rather than us making some educated guess on where we think we need to measure these things. And you can have less bias in assigning finding these critical windows, for example. That's just an important tool that's come along. And then we also measure different environmental pollutants like nitrates on the left-hand side and fine particulate matter on the right-hand side, both of which come largely from traffic-related pollution in these communities. And you can actually see that it's important to know the timing, because where these circles are showing you the effects that are significant in relationship to now not asthma, but again, lung function in these kids, there are different windows. So there are different processes during those developmental stages that are being disrupted by these chemical pollutants. And you need to understand that when you start to try to put this together in a much more comprehensive way. So we're learning a lot from these types of studies, but we're limited by numbers and how much we can do in putting it all together in an integrated way. What else is, why do we bother to measure both social and chemical stressors? Why do we spend so much time doing that? Well, we're glad we did, because you don't always only see independent effects of these two exposures, but you see that stress enhances the effects of the chemical toxins. And that if you didn't look at those things together, you really couldn't start to assign the risk groups. Here in this graphic, you can see the upper right-hand box with the circle around it. It was really boys born to women who had both high level of air pollution exposure in this window, as well as high levels of psychological stress measured on that negative event scale. So you start to see you need to look at higher order interactions. Not only that, you have to look across multi-levels, this complexity science. So we measured both air pollution exposure in these communities from traffic. In this case, we're looking at black carbon, a different measure of that air pollution. And then we also measured community violence exposure. So stress at the community level, not just the negative life events the individual is experiencing. And even when you control for those individual level factors, the community violence had a similar impact in magnitude of effect on wheezing, respiratory illness in these kids as the traffic-related pollutant. Traffic-related pollutant we've always been very concerned about, but not so much the community-level stressors. So we learned a number of principles of life course research through these initial cohort studies. Early events may be as or more important than later events, given that we know what we know about biology. They're going to be critical sensitive windows of development that we need to understand, that time factor. We need to understand how risk factors accumulate over time. Social and chemical exposures interact, so we need to be comprehensively measuring things across these different components. Need to consider multi-level exposures, not just the individual, but where you live matters, place matters, etc. And we see host differences, and sex in particular mattered in what we were looking at. So all of this just has brought us scientifically to knowing that we just need to change our perspective to really design studies going forward that can really advance this work even more than we have already. And we can look through a lens that considers both the physical and social environment and across these multiple levels. And the telescope is over time, understanding it over the course of these developmental windows. Now, we spend a lot of time looking at risk factors, and we realize we also have to understand resiliency factors if we're really going to have this translate into interventions and prevention strategies, which we all want this to translate to. And we want to understand that it's the balance between the risk and resiliency factors that contribute to what trajectory you're on relative to your health over your life course. And that's shown and talked about in the reference that's shown on the right side. So how did we start to do that? Well, we had measured a couple of things that we could start to look at as being levers to pull and help with that. We manifested that most through some of the biomarker data that we looked at. So we also want to do, we want to show how does the external environment get biologically embedded. We look across a number of these systems. We're going to talk about it in a reductionist way, but really as the rainbow on the right-hand side of the prism here shows, we really want to get to where can we integrate and understand how these systems that talk back and forth to each other ultimately influence our outcomes that we're interested in. So let's just look at one dosimeter of toxicity that you can look at as a biomarker. Telomere length is often talked about as an indicator. And if you have shorter telomeres, it's typically a measure of more toxic environmental effects over time, and that it has poor health outcomes linked to it. Well, it turns out that starts even in utero, and telomere dynamics in early development are largely determinant of that relative telomere length over life, and that a number of factors starting in utero can have impacts on it, particularly those that affect things like oxidative stress. So we've shown, and we just published recently a paper using that same air pollution measure over time and the data-driven methods to identify who's at risk. We saw that air pollution was associated with shorter telomere length, particularly in boys. But on the left-hand side of this graph, we also took into account the nutrition data that we had in these mothers. And if mothers were able to have high intakes of antioxidants during pregnancy, you could completely wipe out and mitigate the effects of these other toxins. So that's one potential lever. Another is our social scientist colleagues and our psychology colleagues tell us that social relationships can actually be a buffer against toxic effects of psychological stress, but also these other chemical toxins as well. Well, what relationships are important? Well, that may differ depending on the time in the life course you're looking at, but certainly during early development, which we were looking at, infancy and early childhood, human stress response and those key regulatory systems are really under strong social regulation. And it's your caregiving environment that is particularly central and potentially can buffer the effects of these other toxins that you might be exposed to. So it might be a resiliency building factor. And we've indeed showed that even if you have disrupted hypothalamic pituitary adrenal axis cortisol functioning, which is linked to higher increased risk of our wheezing and respiratory outcomes. If we can show that sensitive caregiving is maintained in that environment, it mitigates these effects. So I kind of think about balancing the risk and resiliency factors as this picture of the train switch yard where someone is trying to make sure things don't get off the tracks or crash. And nutrition and social supports are two levers that we've identified that can actually be those resiliency factors, push people back onto that more optimal developmental trajectory and track and on to better health. Of course, if you go back to the life course strategy, and here we're thinking of lung development, the earlier that you identify the switches and the levers and pull those and intervene, the more impactful that you can be. And again, that is why life course and understanding developmental windows is so important. Now, how do we scale this up? I mean, just in what we've learned and a little bit I've shown you in the cohort work that we've been doing, we see it's much more complicated than we might have first thought about. Well, we're going to measure all these things and we'll look at one at a time for independent effects, maybe an interaction here and there. We have a sample size that can accommodate us looking maybe at a 1- or 2-way interaction, maybe a 3-level interaction, but you can start to see this gets very complicated very fast. And this notion of the exposome, which is on scale of how we think about genomics today, where we're not just measuring one gene, but looking at GWAS or genome-wide. We want to look exposure-wide. And we have a particular challenge here because we always have this time dimension as well. And we need to, in the ideal, what the exposome is to measure the totality and understand the impact of the totality of exposure from preconception to death on the types of health trajectories and outcomes that we're interested in looking at. Now, this isn't a new idea. It's been around since Christopher Wilde first proposed it in 2005. And it's been updated over time, as you can see with the references in the lower right-hand corner. But the exciting thing now is we actually are poised. We have the tools and the computational science to really do this in a major way right now. So it's a very exciting time for environmental health research. We liken it to where the genome project was, and there was the decade of the genomics research, and now that we're heading into the decade of exposome research. We can embrace the complexity, searching for these gene-gene and gene-environment interactions and environment-environment interactions in a significant way with the tools that are emerging. So how do we embrace the complexity and incorporate exposome into translational science? Because that's what we wanted to do. We wanted to translate to health. There's a review article here that walks you through a lot of the things that we're going to say very briefly here. And again, it's to keep those people on track is the idea, to identify signals and networks that can tell us what effective strategies or therapeutic interventions might we have to help people. Well, we need to take advantage of the emerging technologies that allow us to do highly temporally resolved exposure assessment with low burden and reasonable cost. And there are a lot of wearable devices and smartphones, smartwatches, and some of the geocoding that I showed you to do that in a very significant and scalable way, where we can do it across the country, for example, if we had the data sets to do that. And then we could really embrace the complexity of what begets asthma that we're starting to see and starting to picture and wrap our head around, as we show here in this graphic. Other things we can do to move exposomics forward is to understand the complexity of the human exposome. We need to adopt analytical strategies and study designs that incorporate integrated omics approaches, omics, not just one biomarker at a time, but to look across these different platforms, proteomics, microbiome, metabolomics, epigenomics, et cetera, and learn how to integrate those. And we have the tools and the computational skills in a significant way. We also need to adopt analytical strategies and study designs that incorporated untargeted measures of exposure, so we can look at the things we don't even know yet, and not just focus on the few that we do know. And the next important step, and you're going to hear about this more in Dr. Emily Oken's talk, is integrating data across individual cohort studies. So we want to accelerate our ability to look, and if we start building a cohort right now, starting in pregnancy and following over the life course, it's going to take a huge amount of time and it's going to be extremely expensive. But there is a program, the ECHO program, that you're going to hear more about through the NIH, that has asked investigators like the cohorts we talked about here, and merging them with other cohorts across the country so that we can accelerate standing up this cohort that stretches across the life course, not just focusing on where I was talking about early life, for example. We also leverage other resources that have been stood up to help us move the exposomic work forward. The Human Health Exposure Analysis Resource, the HERE program, funded out of the National Institutes of Environmental Health Sciences. This is so investigators, if you have an NIH grant, you can apply to this program, and if you have samples that can be run to do some of these exposure assessments and untargeted analyses and biological response analyses, you can apply, and with no cost to you, you can actually work with these different lab hubs that are shown here and get these different steps done. Now, I showed you the link here too, where you can find how do you apply to it, what's really available through these labs, and I encourage you to go look for that. Now, you might be feeling a little overwhelmed. I want to reassure you, we do not need to measure it all to make significant progress on the exposome. So you do not have to be able to measure every possible exposure over the entire life course to make significant steps forward. Measure as much as you can and try to take advantage of some of these tools that allow you to do so in a major significant way. And then utilize these other resources here, take advantage of things like this integrated harmonized cohort, ECHO, or other tools like that to make these steps forward. Because looking ahead, when I think I start earlier, because it's easier to build strong children than to repair broken men. You think of that picture where do you have the most bang for your buck to put those folks back on a positive health trajectory? And this is just showing sort of the broad view of how we see this all integrating. So thank you very much. It's been wonderful to be able to share with you my excitement about the potential of exposomics in this context, and you'll hear more about how this can play out in the cohort studies beyond what we've talked about here.
Thank you, Dr. Wright. That was a fantastic presentation, and I really appreciate you also highlighting the importance of resiliency and positive benefits, not just negative stressors across the life course. So our next speaker will be Dr. Emily Oken, and she is a professor in the Department of Population Medicine at Harvard Medical School. Dr. Oken, I will go ahead and turn it over to you. Thank you.
Good morning, everyone, and thanks so much for the organizers for the opportunity to present the work that I and a very large team of other people have done standing up a cohort called Project Viva. So we've been asked to give kind of a how-to talk about how you grow, maintain, and expand a longitudinal life course cohort. And also thanks very much to Wright for that fantastic talk, really setting up the context. And as you'll see, it's very relevant to sorry, it's not letting me change my screen. Okay, there we go. So I'm going to talk a bit about how to build a longitudinal cohort, but I just wanted to frame it in the context of the talk that we just heard, which is why cohorts are so important to understanding life course conditions. And I think that those of us who know kind of the foundational science understand that, of course, we can often get the most strongest evidence for causal relationships if we can do a randomized trial. But we also know that trials are expensive, they're complex, they're difficult to maintain. And I would say one of the larger limitations in the context of life course health is that really trials allow us to examine maybe one or a couple of exposures at a fixed dose at a set time period. And when we reflect on the context that we just heard in Roz's talk, that exposures can interact, they can co-occur, they may have different windows of susceptibility, we really need to look to cohort as the foundational substrate for the importance of research into life course origins of health. So this is why a cohort like Project Viva was set up, and I'll give full credit to Matthew Gilman, who is my predecessor in sort of establishing, coming up with the idea and establishing a cohort like Project Viva, which has now been going on for 20 years, amazingly. So Matt and colleagues enrolled women who are receiving prenatal care at a multi-specialty group practice in the greater Boston area in Massachusetts, all of whom were pregnant with singleton infants, and there were over 2,100 births into the cohort from 1990 to 2003. So 20 years later, we have a cohort that continues to include over 1,500 mother-child pairs who remain enrolled, which is a tremendous accomplishment, mostly, I would say, on the part of a terrific research staff who do the groundwork of motivating participants, contacting them, finding them when we lose them, digging through all kinds of resources creatively online, tracing friends and family contacts that they gave us. And so I would say the most important part of a cohort study is a fantastic, dedicated, and motivated staff. And we are fortunate to get people who come in already terrific, but I think that one of the things that we really try to do is help them understand the science behind why something like retention is so important, and how if we don't have strong retention of the cohort, we first of all lose the opportunity to study later outcomes. We lose power to study more rare outcomes or exposures, and we can experience bias in the cohort because we may have differential follow-up. And then I'd just like to give a special credit to one of our staff members, Alex Shostak, who helped with the presentation, which also goes to show you that having a strong, steady staff is helpful in many ways. So this is a kind of high-level representation of the kinds of data that we've collected longitudinally in the cohort. Each of these lines going horizontally reflects one study participant. And so you can see that we recruited on the time axis along the bottom between 1999 and 2002. And then as we follow participants along, we've collected data from different kinds of visits. So, starting at enrollment, we have the dates of birth that are in the green dots, and those we collected by way of medical records. We have had in-person visits with moms and kids. In fact, not represented here is the in-person visits that we did with moms in the clinics at the end of the first trimester at enrollment, and then again in the late second trimester when we collected questionnaire measures, interview measures, and also were able to piggyback on the clinical blood draw to collect additional blood samples for research data collection. So then we saw many of the moms and infants in the hospital at the time of birth. So we were able to collect some measurements that are not really easily obtained from medical records. So for example, infant length, which is very difficult to measure accurately, infant blood pressure, and some additional questionnaire measures about late pregnancy. And then other in-person measures have really tracked with the different developmental life stages. But as you can see, these blue dots sort of line up very nicely longitudinally at birth and then start to spread out with subsequent visits in infancy, early childhood, middle childhood, and adolescence. We're currently conducting our 7th in-person visit, I think, which is at mid to late adolescence as the kids are about 17 to 18 years old. And then in the interim, we collect annual questionnaires, which originally were mailed and now increasingly are online digital questionnaires. And then in addition, we collect biosamples at all of our in-person visits reflected by the red dots. So I sort of like this high-level picture because it really gives you a sense of just how much data is involved longitudinally in following a cohort of this size. So the focus of the cohort from the beginning has been the conditions that are the major health problems that affect children. So initially it was really around pregnancy complications and birth outcomes. And as we followed the kids over time, the main streams of outcomes have included growth, cardiovascular disease risk factors. So we have collected these in a variety of developmentally appropriate ways, starting in infancy with just weight and length measurements, and again, highlighting the importance of research measures, blood pressure. Over time, as the kids got older and more tolerant of blood sticks and fasting blood collections, we've been collecting fasting blood. In addition to the anthropometric measures, which we collected in younger children, as children reached middle childhood and were more able to hold still, as you can see represented in the bottom bubble, we started collecting DEXA measurements on the children, which give us information about body composition as well as overall size. A second stream of outcomes, which Roz mentioned is a major stream that's important for child health, includes asthma and atopy. And again, early in life, we really focus on questionnaire measures. As the kids have gotten older and can participate in more sophisticated measures, we collect breathing measures, including spirometry before and after albuterol inhaler, and multiple measures of allergy, including blood levels of IgE and questionnaire measures and measures of food allergy. And then the last stream is really focused on neurocognitive development. So all the way back into infancy, we have measures of visual recognition memory. And then as the kids develop, we look at everything from IQ to behavioral measures to measures of autism diagnosis, but also autism-related traits such as social cognition. In addition to these outcomes, we really have a pretty expansive view of the kinds of environmental exposures that these children are exposed to. So we think about everything from diet, traffic-related pollution, weather, infant diet, maternal socio-demographic factors, toxic exposures like smoking, and also environmental exposures that are not toxic, like exposure to nature and trees. And again, one of the real reasons that we think cohorts are such a rich source of foundational knowledge in studying life course origins of chronic health conditions is that in one analysis, we can look at something like cigarette smoking as an exposure. In another analysis, we might look at it as a covariate or maybe a confounder or possibly even a mediator if we're thinking maybe of stress causing smoking, resulting in outcome. And then we can also look at these as we sort of move to a more whole exposome related approach, think about multiple exposures in combination. And so this is really the tremendous richness of a cohort study, but of course it adds lots of complexity. And so in order to get all these different kinds of data, we really need to be creative and broad in our collection of data from a variety of sources. So at in-person visits, we collect all kinds of research measures, and we constantly appreciate the tension between, you know, trying to collect everything that we possibly can and not overwhelming our participants. So of course, they get a little more tolerant over time, but we currently are conducting with our older adolescents about a 3.5-hour visits with both mom and kid. At those in-person visits, we collect all these outcome and covariate data that I mentioned by a combination of research measures, also interviews and questionnaires, and then the questionnaires can be fortunately now conducted online as well as in person. We get consent for medical record, and it's a large project to abstract medical records over time. We've also increasingly been using monitors like actigraphs or Fitbits to monitor things like physical activity and sleep. We have address data on all of our participants from every time point since enrollment, and so we can look at the kinds of geocode link data that Roz was discussing, things like particulate pollution, nature exposure, traffic exposure, things like that. And of course, all the variety of biospecimens that we collect, which include not only blood, but also hair, urine, dust from the child's bedroom. And so we've gotten quite creative with all the kinds of things that we can do with this breadth of data sources. So, so that's the sort of high-level picture. And I will say that, you know, as I think about giving a how-to talk and holding a cohort, it's, you know, it's a lot of kind of mistakes made and lessons learned. We clearly did not do everything right all at the beginning, and we've learned a lot of ways to fix things. So, so one of the challenges that of course, we recruited everyone from the greater Boston area, and they were all within local distance when the cohort started, but people move all around the country. And so one of our greatest challenges has been that we want to maintain the cohort and we want to keep them going however we can. So, you know, we need to go find them. And we started a Viva Across America program where we actually do study visits in the pre-COVID era, of course, and hopefully we'll get back to at some point, going to sort of high density places like Seattle and Northern and Southern California, Texas. We've had RAs go to Florida. We had one RA willing to take advantage of her vacation at Las Vegas and do a study visit with the one participant who was there. So really being creative in how we can not only you know, continue to maintain the cohort remotely and also use the more advanced tools that we have for remote data collection to maintain them even if we can't see them in person. This is really not a research talk, so I'm really not going to talk about the wonderful breadth of research that we've done over this time. We have well over 250 publications, I think, by now, but just to make the point that the interesting questions evolve over time. And so, of course, now we're interested in COVID. That wasn't even something anyone was thinking about a year ago. And those kinds of things continually evolve. And so we need to be flexible and innovative as we write our next grant and design our next questionnaire. I would say the biggest challenge has really been, you know, something that works for retention and maintaining interest in a cohort of moms with young infants is very different than we need to do to maintain a population of young adults and older adolescents. And so kind of as our children become adults and can consent and decide whether or not to participate themselves has really created a lot of challenge for us. It helps that many of our study staff at this point aren't that much older than our participants. And so they, I think, can really provide some good suggestions to us. And we've also asked our participants what they want us to do, and I'll show some of those data. We have a tremendous breadth of data from all of these measures on all these people over all of these time points, and organizing and sort of remembering where the data are, accessing it, and making it clear and available to people who want to use our data is an ongoing challenge. And then, of course, just in the current context of COVID, but there have been other challenges, maybe not quite as large, but we and many others have had to kind of shut down research. And so how can we get creative about still maintaining our cohort by remote measures and engaging participants in the kinds of questions that we answer is something that we're just evolving to. So, of course, we do provide incentives, and we think that this isn't the main reason that people participate, but they, I think it helps. And so our cohort maintenance has followed sort of we followed a variety of approaches. When the kids were, when the moms were pregnant, as study incentives, we gave them socks that had the study logo on the sole of the sock, so that when they went to the delivery room, the the clinician delivering them would remember that they were a study participant and collect cord blood. When they were babies, we gave them baby books and word books, and now we just give them cold hard cash because that's what works best with teenagers. But we also give them feedback on some of the measures that we do. So for example, we give each of them a personalized scan of their DEXA, and for the moms that includes a reading of their bone density, which is helpful clinically. We maintain a cohort with regular newsletters, ongoing emails, and we have a website that we are trying to keep up to date to really promote active engagement. And as I mentioned, we really have had to adapt the way we do study, not only in sort of flying around and doing study visits around the country, but also how we communicate with people. You know, even email is just not something that adolescents do. And so we move to text, we move to other platforms to maintain participation and contact, and really keeping up to date with our consent. So we've gone from paper consent to now e-consent so that we can get appropriate informed consent even for people who are remote. So this is just an example of a project that we did to try to really understand what is keeping people engaged in the cohort. And it's been so interesting to hear from the participants themselves, who are really the most important voice. And it was nice to know that they really were mostly engaged around altruism. So both the moms and the teams, the primary reasons for engagement were wanting to help science or wanting to be part of something important. I would say the kids were a little more likely to answer that they were participating because they were reimbursed. And you know, so we've gotten some quantitative measures, but also qualitative measures of really what is helpful. So it's all about participating in the science and the sort of time invested. The interesting thing also is that we're so worried that we're just asking too much of them, but most of them answered that the amount of time that we ask is actually about right, or even maybe not enough. So that's very reassuring to us. So learning from the comments has really been quite informative. And then our favorite feedback on a recent survey that we got, where we asked for any other comments, was a participant who said, "You guys are doing a great job. You deserve a tall glass of lemonade and a ginger cookie." So great endorsement. And then I'll just end, being mindful of the time, I'll move forward a little bit to say that we can learn a tremendous amount from one cohort, but there's really a lot that we need to, a lot more that we can learn from combining cohorts. So an example of a research question that we might ask in Project Viva is the example of prenatal smoking. And we know that smoking during pregnancy is associated with lower fetal growth, but interestingly, we've seen that it also predicts overweight in older children and adolescents. And so as we follow these kids longitudinally over time, the children exposed to prenatal smoking are heavier and with higher risk for obesity. And so we can generalize that beyond just one cohort to do a meta-analysis and look at multiple cohorts, and we see very similar associations, which is helpful to the extent that they might have different confounding structures. And we can also, using meta-analytic data, really get precise estimates of dose-response effects. So the more smoking, the higher the odds of overweight. We can also, in hearkening back to what Roz mentioned, look at combinations of exposures with a cohort. But really the strength of this kind of work is in combining multiple cohorts. And so this National ECHO program has brought together 72 cohorts across the US, representing, I think, 45 states as well as Puerto Rico. So really representing a breadth of and diversity of ages and geography and race and ethnicity. The outcomes of interest in ECHO look familiar. They're actually quite parallel to those that we've studied in Viva, with the addition of a positive health, which is really about well-being and not just the absence of disease, but doing well, which is really what we all want for all of our children. And you know, with this tremendous amount of data, we have to make decisions about what's important. And so I like this framework for prioritizing analyses in ECHO, that it really is the golden intersection of what investigators are passionate about and what's feasible to do, but also really what's going to inform policies, programs, and practices. So I just personally would like to thank my funders for all this work, without whom we couldn't do it, and especially to thank the team that I work with, because research is 100% a team endeavor. So thanks very much.
Thank you very much, Dr. Oken. Our last presentation is going to be given by Dr. Maureen Black. And Dr. Maureen Black is a professor of pediatrics at the University of Maryland and a distinguished fellow at the RTI International. So Dr. Black, I'm going to go ahead and turn it over to you now. Thank you.
Greetings. Thank you for joining. This is Charting the Life Course, Emerging Opportunities to Advance Scientific Approaches Using Life Course Research. My specific topic is From Science to Policy. I am Maureen Black. I'm a professor at the University of Maryland School of Medicine and a distinguished fellow at RTI International. So the objectives is we will talk about advancing policy perspectives in Life Course, and we use a US example from Children's HealthWatch and a global example related to nurturing care. A Life Course perspective illustrates that life begins prior to conception, continues through birth, early childhood, adolescence, adulthood, and into the next generation. This is if everything goes well. But what happens is disparities that start very early in life can alter the life course. And as you see illustrated by the red dots, the disparities can increase over time into the next generation and creating chronic health problems and limited economic potential. So the goal is to look at how we can reduce the disparities. What this slide illustrates is that environments across the life course an impact through epigenetic changes, the health and growth and learning and academic performance and behavior and emotions throughout life. What's key in this is to think about the timing and the quality of the environments and how they shape a child's future potential, because we can alter the environments to enhance or to promote children's potential. These illustrate growth charts, and so we'll just look at how growth occurs very early in life. These are for girls and boys from birth to 24 months. And you can see the slope of the growth that happens very, very steep early in life. If we look at brain development, we also see very rapid brain development early in life. You can see on the x-axis is represented in months. So it starts at conception, goes up by months to birth, up to age 12 months, and then continues on in years. I've drawn a box around the first 1,000 days. That represents from conception to age 2 years. That's 33 months multiplied by 30, and you have 1,000 days. There's a tremendous amount of development that takes place during that first 1,000 days. Synaptogenesis occurs, myelination occurs, and it's very exquisitely timed, so that during this time, children have very specific nutritional and health and environmental needs. If we look at threats to children's development, one that I'll talk about today that's a global concern is food insecurity. This is defined by a lack of consistent access to enough food for an active, healthy life. It's a household level definition. On the triangle illustrates, this is from the USDA, United States Department of Agriculture, that for most of us, it's high food security. And then there's marginal food security. And then it's a much smaller number that are low food security or very low food security. But families, households that are food insecure have risks both for caregivers, and these are things like depression and anxiety, and risks for children. And these are serious health complications or compromising their growth and development. I'm part of a collaboration that monitors the impact of policies and economic conditions on the health of low-income children. This is called Children's HealthWatch, and I'll give you two examples of linking science and policy that come from Children's HealthWatch. We represent 5 medical centers throughout the US. Each medical center serves low-income communities. They are in Baltimore, Boston, Little Rock, Minneapolis, and Philadelphia. We work by having ongoing interviews with parents of children under age 4 who bring their children to the emergency department or primary care. We refer to them as a sentinel sample because they're out front with issues regarding their children's health. We have a data set of over 65,000 children. We're funded primarily by grants, foundations, and donations with a headquarters at Boston Medical Center. It's noteworthy that in addition to an executive director, we have a director of policy strategy so that we keep our eye on how our work relates to policies. This is an example from the Supplemental Nutrition Assistance Program. This is how one can protect against food insecurity or alleviate some of food insecurity. These are food stamps, and it's referred to as SNAP. So we know there's evidence that that SNAP, or that food stamps, protect against later metabolic syndrome. And what we see is that SNAP is actually a work support. 55% of households receiving SNAP have earned income. The eligibility for SNAP is very complex. It's based on a formula that fluctuates based on work patterns. And what we also know is that SNAP has very close federal scrutiny. That means that our federal legislators look at SNAP very carefully, and it is often under a microscope. So the goal of this project was to examine if the loss of SNAP relates to food insecurity and to children and caregivers' health. So we have a large data set and of cross-sectional surveys. So we identified, at this point, we had a pool of 41,000 families. So how do we eliminate or reduce selection bias? So we chose families who either currently or in the recent past had received SNAP. So we know that they're eligible. And we chose families where there was a working household member, and we eliminated a number of confounders, or adjusted for a number of confounders. So our analytical sample in this case was a little over 8,000. You can see the consort diagram on the right. And what we found is that the loss of SNAP, well, we'll see what we found. What this shows in the red box represents percentages. So in our sample, we still have a quarter who are food insecure and 30% whose housing is unstable. This is a very, very high risk sample. So we're asking in this sample if they have had reductions in SNAP or cutoff of SNAP, has that related to children's health or to caregivers' health or to economic hardships at a household level? And what you find is that in many of the areas that we looked at, the answer was yes. So for those who had a reduction in SNAP or a cutoff, that there were adverse economic and health indicators. So from this, we did a policy brief and we did a paper in Health Affairs. Another example is in 2015, there was civil unrest in Baltimore. What happened is that a young man by the name of Freddie Gray was arrested by Baltimore police officers, and in the police vehicle, he sustained neck and spinal injuries and subsequently died. So there were protests citing racism and social injustice and violence throughout the city that erupted. Well, this is a map of Baltimore, and the red stars indicate where the violence occurred. The green dot in the middle is the University of Maryland Medical System. So we were in the middle of this. And the families who receive services, who live in these communities, bring their children to the University of Maryland. We collect data all the time. So we had data before the incident, during the incident, and after the incident. So the question was, how does exposure to civil unrest relate to maternal and child health? So we were able to look at the data the entire year before the event occurred, and then we could look at it in the red box during the immediate aftermath of the event, and then we could look at it after the following the event. And what we found is that the prevalence of maternal depressive symptoms in the communities varied by their proximity to the event, to the unrest. So in the blue line, you can see those who lived in an area proximal to the unrest, and in the pink line, they lived more distally. So the rates of maternal depressive symptoms, the prevalence of maternal depressive symptoms increased in the proximal communities. So they increased up to 50%. So we learned that civil unrest exposes caregivers of young children. These are very young children. They're 3 and under, exposes them to depressive symptoms. And we had a separate survey that showed that it was a disruption to family routines and safety that was associated with their concerns and with their depressive symptoms. So this obtained media coverage, and we worked with the Baltimore City Health Department regarding these findings. I want to show you a bit about global child development, so we'll switch gears to a different topic. There's been very limited attention and investment to children's development from a global perspective. So there have not been population-based measures and no estimate of need from a global perspective or a way to monitor. But yet we know that children under age 5 are clearly at risk, that there are approximately 149 million children who are stunted, so their growth is poor, who are wasted, meaning that they're seriously underweight, and 40 million overweight. So what UNICEF tells us is that 1 in 3 children globally throughout the world is not growing well. The little bit of history, in 2003, the Lancet had a series of papers that looked at where and why are 10 million children dying each year. That was part of the child survival movement, and there were many investments throughout the world to prevent children dying. So what happened between 2003 and 2015, which is 12 years, the decline in child mortality went from 10.8 million to 5.9 million, a 53% decline. Amazing improvement that happened in 12 years throughout the world. Our question was, what happens to children who survive? We were thrilled that many more children were surviving, but the question is, what happens to these children? In collaboration with a number of partners, we produced 3 series of papers on early child development in 2007, 2011, and 2017. These were published in the Lancet. The papers addressed both science and policies. The science looked at the evidence from the first 1,000 days. And what that showed us is that investing in the first 1,000 days then had an impact on the life course through childhood, through adolescence, through adulthood, and into the next generation. We also learned that a system called nurturing care was the way to invest. And I will show you that in the next slide. From a policy perspective, we learned that over 250 million children, or 43% of the world's children under age 5, were not reaching their developmental potential, primarily through poor nutrition and lack of early learning and early stimulation. We calculated the economic effect of doing nothing, of just staying where we were. And what we learned is that it would have a negative impact on over 25% of countries' gross national product, which no country can afford. And then we looked at how investing in early child development could impact the Sustainable Development Goals. This is nurturing care that I referred to, and it promotes early child development by integrating 5 components that children need. Those are having good health, having adequate nutrition, having responsive caregiving, someone to care and look after you, having opportunities for early learning, and having security in where you're living and safety. So these are all necessary. They work together in a system. No one of them is sufficient, that they are all necessary. So how does this happen? We say this is what the child's brain expects and needs, and this is where it comes from. These are enabling environments that start with the caregiver and with the family, extend to communities and services, extend beyond that to enabling policies, and ultimately extend beyond that to climate and to things that impact the world, such as pandemics. So we had a recent publication in The Lancet that looked at children surviving and children thriving and recommended re-envisioning UNICEF's conceptual framework of malnutrition to incorporate some of the principles of nurturing care so that there's attention on the 5 components of health and nutrition and responsive caregiving and early learning and safety. We're also working on what's called the global scale of early development, which is a measure that directly measures early childhood from children who are under age 3. This is, we've harmonized data from 51 cohorts in low and middle income countries, representing over 73,000 children. So in this large harmonized data set, we're in the process of developing a population-based measure so that we can then estimate how countries or regions of countries are doing in terms of early child development. This will then enable countries to put policies and programs into place and evaluate how they are doing, or to identify areas of countries that are in need of additional assistance. This is an effort that is led by the World Health Organization and funded by the Bill and Melinda Gates Foundation and a number of other partners. What this represents, the circle around it, is the Sustainable Development Goals. There are 17 Sustainable Development Goals, and the target for achieving these is 2030. They are very broad and extend from alleviating poverty to gender equity and many, many things in between. And what you can see in the center of the circle is nurturing care. So what this says is this begins with ensuring that our children are healthy and well, and that they can have a life course where they can contribute to society and contribute to the Sustainable Development Goals. So in conclusion, we see that life course is a policy relevant topic. And how do you ensure that your work is relevant and it can inform policy? Some of that is being aware of current issues, being aware of what communities are saying and communities want. And when an issue occurs, such as the Freddie Gray unrest, that you can use your research to answer some questions that can inform policy. The second one is integrating your research into current issues. So the example that comes with SNAP or food stamps. That's a current issue that people are concerned with, that in our case, the federal government is scrutinizing. And so how can we inform what happens if we cut SNAP or cut this federal safety net program? So then that enables our work to be policy relevant. You can focus on strong science. It's always a good idea to focus on strong science. So to ensure that you avoid selection bias and that you address generalizability, so something that happens in one community that can be generalized to other communities. And then to translate complex procedures and findings using policy relevant language. So what that means is to write to multiple audiences. So of course you publish your work from a scientific perspective with a scientific audience, but also to write it into bite-sized pieces in things like commentaries that can have a broader reach and can translate the science into policy relevant language. So I thank you very much for your interest and all the best.
The first question we were asked is, the biologically relevant exposure window may be unique for each exposure. Could you please share some thoughts about how do you take consideration of exposure timing when studying the totality of different exposures over different periods in the lifespan?
Yeah, that's one of the challenges for sure in doing life course research, because the timing of exposure can be equally as important and sometimes more so than dose of the environmental factor. And it really needs to be taken into account. There are evolving statistical and computational methods to account for both timing effects as well as dose. And it's beyond what we went into for the talk, but do be assured that these methods are evolving, and that's why it's such an exciting time to do life course research in environmental medicine now.
Thanks. And then could you share some thoughts about initiating new perinatal cohorts in the near future? Given that we already have at least 50 cohorts in the US, do you feel that these new cohorts are necessary?
I think they're very necessary because we do recognize the importance of timing of exposure, and that we really need to take a more comprehensive approach to analyzing the biological response networks that we just talked about in the session that we're hearing here today. So you have to biobank samples accordingly at the correct time, or incorporate methods where you can do highly temporally resolved exposure assessment, as we discussed. So that hadn't been done to a great extent in prior existing cohort studies, partly for practical reasons, as well, and cost. But now that we recognize the importance of this, I think that we need to invest in standing up cohorts that can really help us address the science that we now understand increasingly.
And as a follow-up, what do you see the role of the older cohort studies in life course research, the ones that are lacking this biological data you talked about?
Well, there's still great value in pulling together and harmonizing the existing cohorts so that we can ask bigger questions together than any one cohort may be able to answer. We may have overlapping exposure assessment in the context of those so that we can account for a broader range of developmental windows and periods, as well as when we're characterizing the clinical outcomes that we're interested in, or more importantly, take advantage of those cohorts to be able to use the biorepositories and the phenotyping that's already been going on to identify early signals of who might be at risk prior to clinical manifestation, because oftentimes it's too late to move the needle as much as we can when you can catch them in earlier states and put them on a healthier trajectory.
Thanks. And then how do you feel your data sets evolve over time as we incorporate these new science tools and technology? Like, do you have a good strategy to help these cohorts stay relevant over time?
That's a challenge, because you kind of have to be thinking ahead and sort of skating where the puck will go, not where you know it is currently. So part of that is practicing team science. That's all the more important reason for us to not stay in our labs and stay so much in our own area of expertise, but to really engage across scientific disciplines and talk amongst ourselves and push each other to really think beyond our own understanding of what's going on. That has particularly been very, very critical to my advanced thinking and how should I approach life course research. I mean, as I talked about, I'm a pulmonary critical care physician taking care of patients at the end of their lives. And all my research starts in pregnancy, because I realized from my colleagues that that's where the roots began. And then I had to, you know, think about what environmental factors do we think is driving this. And, you know, starting out with traditional physical environmental factors that we clinicians think about: tobacco smoke exposure, nutrition, those kinds of things, to realize that the social environment was such an important part of it, and our emotional responding to our environment and the psychological and physiological consequences of that are equally as important. And in my own work and others are showing that maybe more important, particularly early in life, in driving some of these health disparities that we see over the life course. So I had to learn from many of my colleagues outside of my own area of expertise. I'm always trying to engage people across different disciplines.
Thanks. And then I think we have time for one last question. So you mentioned the importance of including social determinants of health in exposomic research. Do you think that the inclusion of social determinants of health and its long-term effects can be used to help influence policy and improve health for people in future generations?
I think it's critical to include the totality of environment. And if we ignore social determinants of health and social environment, and by that I'm talking about, why does poverty drive so much of the pattern that we see in health outcomes? Why is there worse health outcomes across the board, no matter what outcome you're looking at over what period of life? Lower socioeconomic groups, which tend to be more likely ethnic minorities, immigrants, non-native born in the US, when we're talking about the United States, they're more impacted by adverse health outcomes. Well, why is that? What is it about that experience? So then people conceptualize, well, perhaps it's how you emotionally and psychologically experience that environment. They are exposed to a greater number of life stressors, and they have fewer resources to bring to bear to balance those and keep themselves healthy. So we find in our own work, and many others find, that the social environment is equally as impactful as the chemicals that we worry about and think about, like air pollution, tobacco smoke, and that sort of thing. And not only do they have independent effects from those other chemical exposures, but there's synergistic effects. And only when you start to understand the multiplicative effects of the social and the chemical environment do you really see the full scope of what's going on and what's driving programming of adverse health outcomes, starting right from the beginning in pregnancy, but you could look at that at any stage of life.
The biologically relevant exposure window may be unique for each exposure. Could you please share some thoughts about how you take into consideration exposure timing when studying the totality of different exposures over different periods in the lifespan?
Yeah, so that's a good question. So for sure, we are very interested in exposure during different life course periods and trying to identify sensitive periods of exposure. One way to account for this issue is just to measure things at multiple different time points. And that's something that we certainly do and have interest in repeated measures. Ideally, even for exposures for which something like even daily exposure might be relevant, we can use existing data. For example, when we look at air pollution, we use satellite-based measures as one of our exposure metrics, and that is something that's available on a daily level. And then we can apply statistical methods that account for those very high dimensional data so that we can really identify sensitive exposure windows. But mostly, I would say that we're limited to measures that are collected at discrete time intervals, and we just have to be thoughtful that although we're measuring something at a given time, it might actually reflect long-term exposure, or it might really reflect exposure during that window. So we consider that in our interpretation of the results.
Next question is, could you share some thoughts about whether initiating new cohorts is necessary, given that there are already like 50 plus cohorts in the US?
There are certainly more than 50 cohorts in the US. I would say that in the Environmental Influences on Child Health program, the ECHO program through the NIH, there were 74 cohorts that are included. And many more that were not, unfortunately, included. So I think there are reasons to leverage existing data for sure, and not everyone needs to start a new cohort. And probably like every cohort PI, we have data that we've collected that have never been used, and certainly welcome new collaborators who can use our data and ask new questions of existing data. But I think there is some reason to continue collecting new data because exposures change, especially chemical exposures. Measures evolve, and just the burden of disease evolves over time. So while I don't think, I think we should be judicious, but I don't think that we should stop collecting new data on new populations of children.
Okay. And then next question is specific to the Viva cohort, but how have you sustained funding for that study? Is it all through NIH?
The great majority of the funding for Project Viva has been through NIH, although we've had the great opportunity to work with multiple different institutes of NIH. So that's been the largest foundation of our funding. We've also gotten smaller bits of funding, mostly for secondary data analyses from some other sources like the CDC and some foundations.
Okay, and then how do you feel your data sets evolve over time as new science, tools, and technology change? What is your best strategy you've found to stay relevant over time?
So also another excellent question. It's a challenge. I think we certainly have, we started in the era of pencil and paper questionnaires. And so we, an initial change was evolving to online surveys, direct data entry from online questionnaires right into the database, which also has the advantage of saving a lot of effort of research staff having to enter into a database, as well as avoiding data entry errors and preventing things like double data entry, which is hopefully mostly a thing of the past. But as we move forward, we're really looking at more mobile technology based and even data collection that's more passive, that will allow collection of things like location, using cell phone based data, and other things that don't require as much active participation. Personal monitors, exposure monitors, air pollution monitors, other chemical exposure monitors as some examples of things that we're trying to move into, have done some work, but look forward to doing more, because that kind of thing really reduces burden on participants.
Okay, and I think we have time for one last question. So having these long-term cohorts requires having good relationships with your study subjects, and especially for early life and childhood cohorts, it involves engaging parents. What do you think is a good strategy for that?
Well, I think that we need to be honest with participants when we recruit them about what we expect to be the duration of enrollment and also the amount of time and effort that it will involve for them. I think that this is something that we could have done better with our cohort because we really presented it as a study of pregnant women and children, and as our questions evolved and funding evolved and we were able to follow into adolescence and now even into young adulthood, they're a little confused and somewhat because it was not presented to them as a study that might continue that long. So I think what we said that we would have done differently is to say this is a study that's really focused on maternal child health around the time of pregnancy right now because that's what we have funding to do, but we hope in the future to continue to follow you and your family over time so that we can learn more about health as the child grows up.
Thank you. So you ask how social determinants of health can be used to influence policy and to promote health, particularly for the most marginalized groups of people. I think that understanding that health behaviors are influenced by what's happening in the environment and what's happening in people's social spheres can help us understand that it's not just an individual decision. If someone is late for an appointment, it could be they have to take 2 buses in order to get to the appointment. So it broadens our understanding beyond an individual. And in that case, if you had better bus systems or you had better transportation systems, then they might be able to get there on time or might not be late. So it's just understanding that there are aspects of our social system and our environment that influence individual behavior, and people who are marginalized and may not have so many resources cannot change bus schedules or cannot change some of the things that influence their lives. So if we understand that and put that into policies, perhaps to understand why we need bus systems throughout low-income communities, that it has a broader meaning than for policymakers who may not be using the bus systems. So you asked about how to make research translational. I think the very first thing that one can do is to learn to listen. And to listen to what the issues are that are concerning people. If you're interested in health behavior, for example, to listen to what some of the barriers are to that health behavior or to health actions, and then to try and see how that might fit into the work that you're doing. So that you're starting with what the issues are, not what you might think the issues are, but what you have learned by listening to people, to listening to those folks who are confronted with those issues, and then try and work your work into that frame, because then they'll be more interested in listening, because you can then reflect back on what they've told you and how you relate to them. Too often people start the other way with, let me tell you how I can help you. Well, most people don't want to hear how you can help them. What they want to do is to be able to tell you what some of their concerns are. You ask about how technology has changed research. It's just amazing how it has changed research. So some of my closest colleagues, for example, are literally all over the world. And I can talk to them and look at them and be with them every day if I want. So how do you keep up? You keep your eyes open and your ears open. And you do that by reading or by participating in conferences to see what others are doing or what ideas there are and how you could incorporate your work into that kind of thing. People often ask me about how collaborations grow. So collaborations grow by reaching out and by using the technology. And so that we can, I have colleagues who live literally all over the world and in low and middle income countries. And the technology works there well. In fact, many folks have better cellular service than we do. because they got cellular service. They didn't have landlines, but they got cellular service. So it's just keeping your eyes open and reaching out to people.